The complement system. So let's talk about it. So it's a collection of proteins, which are around 25 proteins, majorly produced by a liver, but they can also be produced by other cells like monocytes or macrophage, epithelial cells, and other cells. And they are circulating in the blood in inactive form, waiting for a trigger, or they are enzymes but inactive, so we call them zymogens, with exception of one protein, which is factor D, which circulates in its active form normally, but the others are in their inactive form, and once they are triggered they will initiate a cascade reaction, which will end up in damaging a cell or microorganism. Basically, they will drill a hole on the surface of a microorganism or a cell and leads to a lysis. But they may also have additional roles in addition to this straightforward function or membrane attack complex formation. They will facilitate clearance of the microorganism, and they also facilitate phagocytosis. You can watch the previous video I have made on phagocytosis, and that is by opsonization, facilitating the process of phagocytosis by opsonization. There is also participation of these complement fragments in B cell activation within secondary lymphoid organs or follicles, and we also have talked about this in the previous video. You can watch that. And they also participate in aggravating inflammatory reaction. You can also watch the previous video about inflammation, the role of these complement fragments as a chemokine. So let's talk about them because they may end up in your exam somehow. By the way, if you are new to the channel, my name is Teddy, and if you like my content, please subscribe to the channel and give it a like. Also share it to your friends or other students who may benefit from the content. How does the initiation of alternative complement pathway differ from that of classical pathway? So there are three pathways or cascades which will end up in activation of the complement system: classical pathway, alternative pathway, and lectin pathway. So what's the difference of alternative pathway from the classical pathway? It requires antigen antibody complex containing immunoglobulin M and immunoglobulin G? No. This is the feature of classical pathway. Endotoxin alone can trigger the reaction or activation. Yes, it can activate because alternative pathway is not dependent on antigen antibody complex, but it can be triggered by binding of some complement fragments with antigens directly, including endotoxin. Endotoxin is one good example. The C1 component of complement is involved. No, in the alternative pathway we don't require C1. It's replaced by other components. And antigen antibody complex containing immunoglobulin A and immunoglobulin E can initiate the activation. Well, technically speaking, immunoglobulin A and immunoglobulin E will not participate in the complement pathway activation. So this is wrong completely. So let's see the answer. B. That's correct. So what's the difference in activation of these complement systems? So there are three pathways: the classical pathway, alternative pathway, and lectin pathway. So the classical pathway is discovered first, that's why it's called classical pathway. But later on, when they do further experimentation, there are other mechanisms of activating the complement system, so they called it alternative pathway. And there is also completely innate defense mechanism, which is not dependent on the antibody, and it activates the complement pathway similar with the antibody dependent pathway, and they call it lectin pathway. So in the classical pathway we will require antigen antibody reaction. Specifically, we will require immunoglobulin G and immunoglobulin M. So why these two? Because they have a binding site for one complement factor, which is called C1, and C1 have three components. It's like a bundle of matchsticks with heads. So it have around 6 heads, which are matchsticks, and this also associated with other proteins, a proteins, a tetramer. 2 of the C1R and 2 of C1S with C1q, which 6 of C1q. So C1 have 3 components, Q, R, and S. So the head of C1q will bind with immunoglobulin G and immunoglobulin M, Fc regions, specifically the constant 2 domain of immunoglobulin G and the third constant domain of immunoglobulin M. So once it binds with immunoglobulin M and immunoglobulin G Fc region, it will initiate the cascade reaction, but it requires 2 Fc regions. So since immunoglobulin G is a monomer in the circulation, it cannot initiate the cascade reaction by itself. It must be bound to antigen, a multivalent antigen. So the 2 Fc regions of immunoglobulin will appear in proximal regions. And for immunoglobulin M, even though it's a pentamer, even though it have 5 Fc regions in a proximal appearance or together, it cannot initiate the cascade reaction because they are not accessible to the C1q in their pentamer or in soluble forms. But when they are bound with the antigen, the binding site for C1q will be exposed and it will bind with the C1q. So once C1q binds with the Fc region of antibody, it will have a conformational change which leads to activation of C1r, and C1r leads to cleavage and activation of C1s. Now this C1 complement factor is active, so the C1s binds with or cleaves C4 and C2. So once a C4 is cleaved, it will create 2 components, a smaller component which is labeled as small letter A and a larger component which is labeled as small letter B. So C4 will lead to creation of C4b and C4a. So you can take A for active component and B for binding. So the binding will remain on the surface of the antigen. If it's a cell, on the surface of a cell, and if it is a bacteria or microorganism, on the surface of microorganism. So this C4b have a thioester domain which preferably binds with sugar molecules which are not found on the surface of human cells, preferably on microorganisms. So it will easily bind and stick on the surface of microorganism, waiting for other complement factors. C1s also cleaves a C2, but in case of C2, the A is larger in size and the B is small. It's exceptional for from other complement factors, and the A will remain bound with a C4b, which already is bound with a membrane of a cell or microorganism, and the B will be released. The B, well, its function is not clear. This bound C4b and C2a, the B is a binding site from a C4, and the A from a C2 is active, proteolytically active. So it will convert other complement factors into its active form. Specifically, it will attract C3 and it will convert it into C3b and C3a. So in classical pathway, C4b to A is a C3 convertase, which means it will catalytically convert C3 into A and B. Again, the B component have a thioester domain similar with C4b, and it will stick on the surface of a cell or microorganism, and the A component will be released. So the A have another separate function, we'll talk about that. But the B will remain bound on the surface of a cell. So this complex C4b, C2a, and C3b is a C5 convertase for a classical pathway. Again, it have a binding site from C3 and C4, and it have a proteolytically active enzyme site from C2, which is C2a. And again, this converts C5 into A and B. Again, the B will remain bound on the surface of a cell or microorganism, and the A will be released. So this is how the classical pathway is started. Once it reaches C5b, we call it a terminal pathway, and it's similar for all 3 pathways, but the difference is in the initiation. So what happens to the classical pathway will not happen in the case of alternative pathway. For alternative pathway, it will not require antibody molecules like that of the classical pathway. So the alternative pathway is dependent on C3 takeover, which means there is also a spontaneous conversion of C3 into B and A. So like 1 to 2% of a C3 protein will be converted into A and B normally. But I told you that the C3 and C4, once they are converted into a B and A, the B component or B fragment have a thioester domain which binds preferably to sugar molecules on surface of microorganisms. But if it remains on soluble form, it will be deactivated by water or hydrolyzed. So if it remains in the solution, it will be hydrolyzed. That's a normal control system. But if it bounds on the surface of a bacteria, if there is a microorganism in the surrounding, it will bind to the surface of a bacteria and it will attract another factor which is called factor B. So the C3B bound on the surface of microorganism will attract factor B, which is a plasma protein. So this factor B will attract factor D, which is active normally. It circulates actively. So it doesn't need activation from other proteins, but it circulates in a controlled environment. So this factor B will cleave factor B into A and B, which creates a confusing name. Like the B will remain on the surface of a cell which is associated with C3B, so it gives C3B BB. So the A will be released. So the name looks confusing. So the C3B BB is a C3 convertase for the alternative pathway, but the classical pathway have C4B to A as a C3 convertase, but the alternative pathway have C3B BB as a C3 convertase. So this will convert more C3 molecules into A and B. Again, the A will be released and the B will remain on the surface of a microorganism. So it gives a complex like C3B BB against C3B, which is a C5 convertase for alternative pathway, and it converts C5 into A and B, and it will have a terminal pathway following that. So this is the alternative pathway activation. But for the lectin pathway, it fairly looks like the classical pathway, but without antibodies and a C1 protein. So for a lectin pathway, it's dependent on colectins like mannose binding lectins and phecolins. So the mannose binding lectins and phecolins will bind to sugar residues on the surface of microorganisms. So the mannose binding lectin have a mannose binding lectin active head, which is going to replace the C1q. It will also have associated proteins like mannose binding lectin, associated serine protease 1, associated serine protease 2 and 3. So it may have 1 and 2 or 2 and 3 at a time. It's not going to have 3, but 2 of them will be associated with mannose binding lectins. So in phecolins, we have 3 main types of phecolins. M phecolin, which is majorly produced by macrophage, or the other name is phecolin 1. We have also L phecolins and H phecolins, like phecolin 2 and 3 respectively. So they can be found on the plasma, but M phecolins are majorly produced by macrophage. So these phecolins will prefer acetylated sugars. However, in case of mannose binding lectins, they prefer mannose. So that's why they are called mannose binding lectin. So this mannose binding lectin will bind with a sugar residue on the surface of microorganisms, which is not available on human cells. And once it's bound, it will have a conformational change, which will activate a MASP1, which is mannose binding lectin associated serine protease 1, which in turn converts or activates MASP2. Now, once MASP2 is active, it will have a role of C1s in case of the classical pathway. Then it will attract C4 and C2 and will convert them and will create C4b to A, which is a C3 convertase. So the C3 convertase and C5 convertase for a lectin pathway and the classical pathway are the same. The difference is the initial step. In case of lectin pathway, there is no requirement of C1 protein and antibody molecule. However, in case of classical pathway, there is a requirement of C1 protein and antibody molecule. So once it reaches the C5, once the C5 is converted, it creates CB and A components. The B will remain bound on the surface of a microorganism or a cell. It will immediately bind with a C6. Then it will insert C6 on the membrane. Then it will attract C7. It will insert more. So it's like hitting it on the surface of a cell until it reaches the cytoplasm. So it will attract C6. It will have short chain inserted on the surface of a cell. Then when C7 comes, it will have a longer insertion on the surface of a cell. And when C8 comes, it will reach the cytoplasm. So technically it have created a pore, but it's not enough for release of content or cytoplasmic content, as well as entry of extracellular content into a cell. So it will not be enough to lead to lysis, but if it's much in concentration on the surface of a cell, it may lead to lysis. So here comes the role of C9. So once C8 creates a pore, a C9 is a stabilizing protein. Like 10 up to 16 fragments of a C9 protein will come at the site of pore formation, and it will maintain large opening on the surface of a cell. If there is no C9, the pore will not be maintained and there will not be a lysis. So this is a difference of the 3 complement pathway. So we said that once the complement proteins are fragmented, they will create a larger portion, which is called B, with the exception of factor 2, or complement factor 2, and the smaller fragment A. So the A is always released with the exception of complement factor 2, which remains bound to the surface of a cell or microorganism. So those released fragments have additional role. So you could be asked about those fragments. So let's see, for example, what is the function of C3A, C4A, and C5A? The fragments produced during the complement cascade. To bind to specific membrane receptors on lymphocytes, triggering the release of cytotoxic substance? No, they are not going to induce release of cytotoxic substances. To increase vascular permeability, induce smooth muscle contraction, and stimulate histamine release by basophils? Yes, they are anaphylaxis. They will aggravate inflammatory reaction, so this seems to be correct. To attach to membrane receptors on macrophage, enhance phagocytosis and the elimination of debris and foreign particles. No, so complex removal is not mediated by the A fragment, rather it's by the B fragment, specifically C3b and C4b will participate in antigen-antibody complex removal or clearance from the body. So this is not the correct answer. So to regulate and degrade membrane cofactor proteins following activation by C3 convertase. No, they are not regulatory proteins. So the correct answer will be B. So these fragments, the A fragments, are called anaphylaxins, and they are potent in inducing vascular permeability as well as vascular contraction and relaxation, which is the initial step of inflammatory reaction. You can watch the previous video about inflammation if you want the details of inflammatory reaction. So these A fragments also have receptors on basophils and mast cells, which will induce the release of vasoactive amines packed within these cells, and which will aggravate inflammatory response. Basically, after inducing this inflammatory response, they will allow, or it will lead to attraction of more white blood cells to the site of infection. Again, it leads to defense mechanism. So one of the innate defense mechanisms by these fragments too. This complement system, since it have a devastating effect, if it's not controlled, it may lead to destruction of self-tissue. So it have to be controlled tightly. So there are regulatory proteins for complement system. If they are deficient, it will have a devastating effect. So the controlled proteins may also end up in your exam. So let's see. A patient is found to have a genetic deficiency of CD59, a regulatory protein of the complement system. What effects would a deficiency in CD59 have on the complement system? So CD59 is one of the regulatory proteins for the complement system. If it's deficient, obviously we will expect some abnormality, but what specific abnormality? Increased activation of a classical pathway due to lack of C1 regulation. No, CD59 is more of a terminal pathway control system. So it's not initiation pathway. So C1 is not the major target. Uncontrolled formation of membrane attack complex or MAC leading to increased cell lysis. This seems to be the right. Reduced cleavage of C3 and C5 resulting in decreased complement activation. Well, it's not reduced cleavage. When we are deficient in regulatory proteins, we will have increased cleavage and increased complement activation system. So this is not right. Impaired phagocytosis due to reduced opsonization by C3b. This is again wrong. So the correct answer will be B. So in here, what are the regulatory proteins and what is their major target? So there are several regulatory proteins which will regulate the complement cascade or activation system. One of the regulatory protein is C1 inhibitor. As the name indicates, its target is C1. Specifically, C1R and S components of a C1 protein will be targeted, and it will inhibit and disintegrate the function of C1R and C1S. So if they are disintegrated and if they are inhibited, there is no conversion of C4 and C2, and the classical pathway and the lectin pathway will be stopped. The other regulatory protein is factor I. Majorly, it targets C4b and C3b, and it's again a serine protease which will cleave C3b and C4b by using another protein which is called factor H, as well as MCP, C4b binding protein, and complement receptor 1 as a cofactor. So this factor I requires cofactors, so it's not going to do its job alone, but its target is C4b and C3b. So if there is no C3b and C4b, there is no continuation of the complement cascade. The other protein is factor H, which is a cofactor for factor I, and it majorly targets C3b, and it binds with C3b and disrupts or displaces BB. Again, we said that we require BB for the alternative pathway. So majorly factor H targets the alternative pathway by itself, and since it's a cofactor for factor I, it may also target the other complement cascades. And the other is C4 binding protein. As the name indicates, it majorly binds with C4 and it stops the classical pathway and the lectin pathway. And the other is membrane attack cofactor protein or CD46, and it targets C3b and C4b. And we also have decay accelerating factor or DAF, and this targets complexes like C3 convertase complexes, which are C4b2a and C3b-BB, and it will disintegrate this complex and stops further conversion of C3 into active forms. And CD59 majorly targets C7 and C8. It blocks C9 binding and prevents MAC formation. So these are some of the regulatory proteins in the complement system. So the other thing is there are also binding proteins for a complement system. So we can classify complement receptors into 4 major groups: type 1 complement receptor, type 2 complement receptor, type 3 complement receptor, and type 4 complement receptor. They also have other names. For example, in case of type 1 complement receptor, the other name is CD35. Majorly, it prefers C3b. It also binds with C4b and IC3b. So IC3b is unstable fragment of C3. So C3 will also be further fragmented into C3d, C3dg, and IC3b. So these fragments of a C3b as well as C3b in its native form have active biological role. One is continuation of the complement cascade and formation of a MAC. The other is by interacting with its binding protein or receptor protein on different cells, it mediates other functions. It could be opsonization, it could also be activation of a B cell in secondary lymphoid organ. So this complement receptor 1 or CD35 is majorly found on phagocytes, neutrophils, B cells, T cells, erythrocytes, and follicular dendritic cells. So since it's found on fairly different types of cells, it may have different role. If it's found, for example, in case of phagocytes and neutrophils, it will facilitate phagocytosis. And in case of erythrocytes, clearance of immune complex from the system or from the blood. So erythrocytes have a role in defense mechanism, since fairly they are large in number and they have this CD35 on their surface, and if they are in contact with antigen bound or covered with C3b, they will clear it from the system using this complement receptor 1. And it also promotes dissociation of C3 convertase by acting as a cofactor for factor I. We have talked that previously. And the other is complement receptor 2 or CD21. So when we talk about cells and organs of the immune system, we talked about this CD21. You can watch the previous video about that if you want the details about that. And it's one of the CD marker which found on the surface of B cells. It can also be found on follicular dendritic cells and nasopharyngeal epithelium, but it prefers C3b fragments. Majorly, it prefers C3d. It can also bind with DG and IC3b, but majorly it prefers C3d, and it participates in activation of a B cell, naive B cell. It serves as a co-receptor for B cell activation. It also leads to trapping and maintenance of antigen in the germinal center. So there are follicular dendritic cells within the germinal center, and they will lead to activation of a B cell. But for that to happen effectively, we should sustain the antigen within the center, and this complement receptor 2 will participate in that. And the other is complement receptor 3, and the other name for this is MAC1 or CD11b complexed with CD18. So when we talk about phagocytosis and white blood cell migration in the previous video, we have talked about MAC1, which leads to stasis of rolling white blood cells on the surface of endothelium. Before they migrate into a tissue space, they will have rolling and stasis. So one of the integrins which participate in the stasis is MAC1. There is also leukocyte function antigen 1 together with that, but MAC1 has that role, and it prefers IC3b and ICAM1. So when it interacts with IC3b, it leads to phagocytosis. when it interacts with ICAM1, it leads to migration of leukocyte into the tissue space for its activation, for its antigen clearance role. And there is also type 4 complement receptor. It looks like complement receptor 3, and majorly it interacts with IC3B. So these are some of the complement receptors and their role in immunity. So hopefully this glimpse summary of the complement system was helpful in your exam preparation. Please subscribe to the channel and share it with your friends. Until we meet next time, see you.